wabarakatuh. My name is Muhammad Razi Zaidi bin Rasidin. I'm from Group 10 and now I will tell about practical lab for digestion of starch and lipid. For the objective, we examine the function of common enzyme in digestion of starch and lipid. And the second objective is we determine the factors affecting enzyme activities. For experiment 4.2, digestion of starch, the methodology is 5 pairs of test tube. We prepare as follows. There is 5 pairs of tube. First pair is A1, A2, which is contain 4 ml glucose only. For the test tube B, for pairs B1 and B2, it contains 4 ml starch only. For the pairs of tube C, contain 4 ml untreated amylase only. Tube D, 2 ml starch plus 2 ml untreated amylase. And for tube E, 2 ml starch and 2 ml acidified amylase. All test tube will shake and leave for 10 minutes. The test reagent added as follows. 0.5 ml of iodine solution added into test tube A1 anti-E1 and well shake and leave for 10 minutes. Observation recorded. 0.5 ml of Benedict's reagent added into test tube A2 E2. Last but not least, well shake and incubated in boiling water bath 100 degrees Celsius for 5 minutes and the observation recorded. All the observation was recorded at table 4.2.
Hi, my name is Nur Hazima binti Jasni AS 11759. So I will be explaining the result of digestion of starch. So the first pair is labeled as A1 and A2 containing 4 ml of glucose. The iodine test result is no change in the color of solution, while the Benedict test is a brick red color of solution form. So the conclusion is iodine test show absence of starch in solution, while Benedict test show presence of glucose in tube solution. So the second pair is labeled as B1 and B2 containing 4 ml of starch only. The result of iodine test is a blue-black color is formed, while the Benedict test is no change in the color of solution. So the conclusion is iodine test show presence of starch in solution, while Benedict test show absence of glucose in tube solution. The third pair is labeled as C1 and C2 containing 4 ml of untreated amylase. The result of iodine test is no change of color, while the result of Benedict test is a yellowish orange color form. So the conclusion is iodine test show absence of starch in solution, while in Benedict test show presence of glucose in tube solution. So the fourth pair is labeled as D1 and D2 containing 2 ml starch plus 2 ml untreated amylase. So the result of iodine test is a blue-black color form, while the Benedict test result is a dark orange color form. The conclusion is iodine test show presence of starch in the solution, while Benedict test show presence of glucose in the solution. So the fifth pair is labeled as E1 and E2 containing 2 ml of starch and plus 2 ml acidified amylase. So the iodine test result is a blue-black color form, while the Benedict test result is an orange color of solution form. So the conclusion is iodine test show presence of starch in the solution, while Benedict test show presence of glucose in the solution.
Alright. My name is Muhammad Afif Ibrahim and I'm from Kuliah 18. So today I'm gonna discuss about experiment 4.2, which is experiment digestion of starch. So for this experiment, the result that we obtain, which is in test tube A that consists of glucose, shows negative result in iodine test and positive result for Benedict test. For test tube B that consists of starch, shows positive result in iodine test and negative result for Benedict test. For test tube C that consists of untreated amylase, shows negative result in iodine test and positive result for Benedict test also. For test tube D, that consists of starch and untreated amylase. Both shows positive result in the iodine test and Benedict test. And last but not least, for test tube E, that consists of starch and acidified amylase, shows both positive result in iodine and Benedict test. So the acidified amylase in the test tube E prevent the reaction of amylase with starch to occur as the amylase is denatured. So the observation that we occur is in iodine test, the solution change from yellow to dark blue if starch is present. And in Benedict test, brick red precipitate can be seen if reducing sugar is present in the solution. So based on the expected result, we should obtain negative result in test tube C and E for Benedict result because reducing sugar is not present in the solution. So maybe the problem that we face may causes from the contaminated dropper of solution or contaminated beaker. So in order to get accurate result in iodine and Benedict test, make sure the dropper and beaker is not contaminated. So that's all from me. Thank you.
For the experiment 4.3 for digestion of lipid, the methodology was as follows. The test tube will prepare as follows. We have test tube A, B, C, and D. But for test tube A, 2 ml of lipase and 4 ml of bile added. 2 or 3 drops of phenolphthalein and 0.2% natrium hydroxide, drop by drop added until the solution turns pink. Next, 3 drops of vegetable oils added and well shaked, and then incubated it at 37 degrees Celsius for 1 hour. For test tube B, which contains 2 ml of lipase and 4 ml of distilled water, 2 or 3 drops of phenolphthalein and 0.2 natrium hydroxide also drop by drop added until the solution turns pink. 3 drops of vegetable oils added and well shaked. The tube incubated at 37 degrees Celsius for 1 hour. For the test tube C, which is contain 2 ml of distilled water and 4 ml of bile, the second step is 2 or 3 drops of phenolphthalein and 0.2% sodium hydroxide, drop by drop added until the solution turns pink. 3 drops of vegetable oil added and well shaked. The tube also incubated at 37 degrees Celsius for 1 hour. For the last tube, for tube D, which is contains 2 ml of lipase and 4 ml of bile, 2 or 3 drops of phenolphthalein and 0.2% sodium hydroxide, drop by drop added until the solution turns pink. 3 drops of vegetable oil added and well shaked. But for this tube, we leave at room temperature for 1 hour. The initial color and the final color of each test tube were observed and recorded in table 4.3.
The result of digestion of lipid. So the first test tube labeled as A containing 2 ml of lipase plus 4 ml of bile. The initial color is brown. Then the color changed from brown to dark green at the final color. So the conclusion is reaction does not occur. So the second test tube is labeled as B containing 2 ml of lipase plus 4 ml of distilled water. The initial color is dark pink. Then the color changed from dark pink to light pink at the final color. So the conclusion is reaction occur. The third test tube labeled as C containing 2 ml of distilled water plus 4 ml of bile. The initial color is brown and the final color is also brown. So that means there is no color change. So the conclusion is reaction does not occur. So the fourth test tube is 2 ml of lipase plus 4 ml of bile. The initial color is brown and the color changed from brown to light green at the final color. So the conclusion is reaction does not occur.
My name is Anis Nadira and I'm going to present on discussion 4.3, which is digestion of lipid. Based on the experiment 4.3, the result that we obtained in test tube A, the solution changed color from pink to light green. For test tube B, reaction occurs very slowly as the lipase will have to digest the vegetable oil without the help of bile salts, which will emulsify the vegetable oil. Reaction had occurred as it can be seen, the shade of pink solution becomes lighter in an hour. This result obtained is almost same as the expected result, with the difference being in the shade of the pink color. While in test tube C and D, the solution should remain dark pink. In test tube A, the solution contains lipase and bile, thus digestion of lipid occurs optimally. In test tube C and D, the hydrolysis of lipid cannot occur. It is because in test tube C, lipase is not present. In test tube D, the result is negative because digestion of lipid occurs slowly in room temperature. For this reason, the optimum temperature of digestion lipid is 37 degrees Celsius. The color of the solution is to supposedly remain pink at the end of experiment, but there was errors that occurs in this experiment causing the experimental result to be green. One of the ways to improve the experiment, we should make sure the dropper and beaker is clearly clean and not contaminated to obtain an accurate result. I think that's all from our presentation on experiment 4. Thank you.